The topic is about the DNA or the blueprint of life. When we say blueprint, it is the design, guide, and plan that can be followed. So DNA was first described in 1953 and it stands for deoxyribonucleic acid. So it is known that the double helix structure was established by James Watson and Francis Crick through X-ray crystallography. But before that, pioneering the study of the structure of the DNA, we have Rosalind Franklin, wherein she used X-ray diffraction and she was the first person to see the structure of the chromosome. But due to her death, so hindi niya na-claim yung Nobel Prize, si Rosalind Franklin. She died because of too much radiation due to X-ray diffraction. Dun sa X-ray diffraction na yun, nakita yung X and X chromosome, which is from a female. And then, through X-ray crystallography, doon naman nakita ni James Watson and Francis Crick yung nasa chromosomes. Remember that chromosomes are coils of DNA. So, doon na nila nakita yung double helix structure of the DNA. So, for the key features of the DNA as a genetic material, the DNA is stored. So, it is stored because it is organized into genes and packaged in chromosomes. So, the genetic material or the traits that comes from or that came from the parents is stored in the DNA. Next, the DNA is replicated. So, the genetic information can be duplicated. So, we can have an exact copy of the trait from the parents to the offspring. It is also expressed, so it has provided with different mechanisms to affect phenotypes. And remember that phenotypes are the traits which are physically expressed. So, in the DNA, kaya nga siya tinawag na blueprint because it is the plan what will be your physical appearance or physical characteristics. Next, the DNA is diversified. Meaning it has diversity, it has the ability to mutate and to produce variation and drive evolution. That is why meron tayong kahawig pero not the exact copy of us. That is why one person is different from one another. There is species diversity. Ayun. So, those are the 4 key features of DNA as a genetic material. DNA is stored, replicated, expressed, and diversified. So, next, we have here the structure of the DNA. To study further on the structure of the DNA, let me discuss its parts. It is composed of repeating subunits called the nucleotide or the monomers. The nucleotide has 3 main parts, which is the phosphate group, sugar, and the nitrogen base. For the sugar, we have deoxyribose, also known as pentose, because as you can see, it is in the shape of pentagon. For the nucleotides, or the nitrogen bases, we have thymine, adenine, cytosine, and guanine, which are classified into 2 classifications, which is purines for adenine and guanine, and pyrimidines for cytosine and thymine. To classify the structure of adenine and guanine, so they are purines. Remember that purines have 2 ring structures and 2 bases, while for pyrimidines, which are cytosine and thymine, it has only 1 ring structure. Para din mabilis tandaan yung pyrimidines na nucleotides, cytosine and thymine, meron silang letter Y doon sa kanilang pangalan. So, the ones responsible for the double helix structure, kaya siya spiral, is the sugar-phosphate backbone. So, for the backbone, it contains, kaya nga siya sugar and phosphate because it contains alternating phosphate. and sugar. Kanina nabanggit ko that this sugar is deoxyribose and also a pentose. Because yung naalala nyo sa organic chemistry, kaya siya naging ganito. This is a cyclic structure. It contains 5 carbons. So, this is also known as pentose, yung sugar niya. Okay, so again, alternating phosphate and sugar that consists the sugar phosphate backbone. Pag tumingin naman tayo dito sa inside ng ating DNA, we have here the complementary bases or complementary nitrogenous base, which is adenine is complementary to thymine, and it is bonded by hydrogen bonds. Also remember that adenine and thymine are bonded by 2 hydrogen bonds, while guanine and cytosine are bonded by 3 hydrogen bonds. Okay, for the next topic we have here DNA replication. So DNA replication is the process by which DNA makes a copy of itself. So it happens during cell division. So we have 3 steps in DNA replication. So for the first step, we have here unwinding. So how does the DNA unwind? So through the help of an enzyme called the helicase, dumadaan dito sa gitna, bine-break down ni helicase, yung enzyme na yon, yung hydrogen bond na nagko-connect between the nitrogenous bases, between adenine, thymine, and cytosine and guanine. So, yun yung ginagawa ng helicase, and that step is unwinding. And remember that DNA is semi-conservative. Pag sinabing semi-conservative, half of the old strand is saved, meaning nasisave yung both strands ng doon sa double helix natin. Then, the next step is, number 2, will be the complementary base pairing. So, kapag nag-unwind na siya, so halimbawa, ang nandito sa kabila is letter A, or the adenine. So, of course, it will have a new complementary pair, which is thymine. So, ito yon. So, complementary base pairing is for the step 2. Next is for the step 3, joining. So, in joining, we have also, if there is an enzyme in order for the hydrogen bond to break down or to break, we have here an enzyme. In order for the hydrogen bond to bond again the nitrogenous bases. So, ibaband niya ulit yung mga, yung panibago namang base. So, it happens in joining. So, nandun yung DNA polymerase. So, if this is adenine, this one is thymine. So, dito naman sa kabila, kung ito yung thymine, ito naman yung adenine. Okay. Ayan. So, that is DNA replication. For the next one, we have here DNA transcription. When we say transcription, it is the process of having a copy of the DNA. It is being transcribed into RNA. So, DNA to RNA, it occurs in the nucleus. So, again, the process of transcription begins when an enzyme called RNA polymerase attaches to the template DNA strand and begins to catalyze production of complementary RNA. So, alamin muna natin, ano ba yung RNA na sinasabi? So, RNA is a single strand, unlike the DNA, which is a double strand. And the sugar that is present in RNA is ribose only, not deoxyribose, unlike the DNA. So, it contains no thymine. It has no thymine, but instead, the partner of adenine becomes uracil. But then, yung partner ni thymine ay adenine pa rin. So, ang nababago lang sa kanya yung thymine, nagiging uracil. So, the difference between the DNA and RNA is that DNA, nandun lang siya sa loob ng nucleus, while the RNA can leave the nucleus because the RNA produces amino acids which is needed by the ribosomes. Okay, so it follows base pair rule and codon sequence. So what is a codon? A codon is a sequence of 3 nucleotides. And it has a complementary, yung anticodon. So the complement of 3 nucleotides. So I have here, for example, so the DNA strand is thymine, adenine, and cytosine. So these 3, itong tatlong to, it is known as a codon. Its anticodon in RNA, or the complement of it in RNA, is A, adenine, and U, uracil, G, guanine. So this is the codon, and this one is anticodon. While for this one, the DNA is guanine, the DNA strand is guanine, cytosine, and guanine. And then for the complementary or the anticodon, we have cytosine, guanine, and cytosine. So that is how you are going to transcribe DNA from RNA. Pag DNA replication, parehas lang sila. Okay, ang ginagamit sa DNA replication is thymine pa rin. But then, if it is for transcription, so ang gagamitin na natin is uracil. For the next one, RNA translation. Ano naman yung translation? Hindi ko sa tinawag na DNA translation kasi yung unang term talaga is DNA transcription and DNA translation. But since we already transcribed the DNA into RNA, ano na yung ita-translate mo? Diba yung RNA na yung ita-translate. So, ang nilagay ko na dyan is RNA translation. So, what happens in RNA translation? So, the molecules will be interpreted into amino acid and it occurs in cytoplasm. Bakit sa cytoplasm nag-occur yung translation? Kasi yung amino acid na mapoproduce niya, kailangan siyang pumunta doon sa ribosomes para ma-synthesize as protein. So, remember that ribosomes are outside the nucleus. Okay, so I have here an example. Yung pa rin kanina, but then it is already translated into amino acid. So, nakuha natin kanina yung RNA, which is adenine, uracil, and guanine, cytosine, guanine, and cytosine. Okay, so we're going to look for, ano yan, methionine and arginine. Saan ang galing yon? So, there is a table. Ito yung table. Okay, so ito yung table natin of amino acids. So, each codon na makikita natin, yung anticodon, each anticodon has an equivalent amino acid. So, we have a total of 20 amino acids. And we always start with the amino acid, which is, we always start with the amino acid, which is methionine. So, kung mapapansin nyo dun sa unang slide, yung na-convert natin o yung nakuha nating RNA is AUG. That is always the start codon na tinatawag. So, hindi magta-translate ang RNA strand kapag hindi siya nagsimula doon sa AUG. So, lahat yon sa eukaryotes, it always starts with the RNA, AUG, with the amino acid, met or methionine. Okay, so we have here itong PHE, phenylalanine, LEU, leucine, leucine pa rin to, next ILE, isoleucine, methionine, VAL is valine, SER is serine, PRO is proline, THR, threonine, ALA, alanine, TYR, tyrosine, and then we have here the stop codon. Kapag tinawag siyang stop or stop amino acid or stop codon, nag-stop na yung translation niya. So, bali wala na yung sumunod na RNA strand. Hindi na siya nasisynthesize as protein kapag nagkaroon tayo ng codon na UAA or UAG. Yung equivalent niya is stop. Next, we have here histidine, then glutamine, ASN is asparagine, LYS lysine, ASP is aspartate, GLU is glutamine, CYS is cysteine, and we have another stop codon which is ito, yung UGA. Next, TRP, tryptophan, ARG, arginine, and again, serine, arginine, and GLY, which is glycine. Ayan. So, ayan naman yung mga sinabi ko na amino acids. Ayan yung complete name nila. Yung nasa isa, nasa kabilang slide is abbreviation lang. Okay. So, ito, kung balik tayo dun sa translation. So, yung AUG, itong RNA na to, hahanapin mo siya dito sa table, which is AUG stands for the amino acid methionine. Ito namang isa, CGC, hahanapin mo rin siya dito sa ating table of amino acids. CGC stands for ARG or arginine amino acid. And then, those amino acids will be synthesized together. So, di ba, strand siya, isang mahabang strand yung ating RNA. So, marami yung amino acid, and those amino acids will combine and synthesize to form proteins na nagagamit natin in energy building. So, we have the three types of RNA. So, for the three types of RNA, we have messenger RNA or the mRNA, transfer RNA, tRNA, and ribosomal RNA or rRNA. So, what is the function of these three? Saan siya nangyayari? Or saan pumapasok itong three types of RNA? So, yung messenger RNA, sa simula pa lang ng transcription, nandun na si messenger RNA. Okay, when we transcribe DNA to RNA, so ayun na yung message, ayun na yung code. So, it copies the information. Kinokopya ni mRNA yung code galing sa transcription. Next is transfer RNA. From the word itself, transfer, of course, it carries or transfer the information na galing dun sa mRNA. Si mRNA, pinasa kay tRNA yung nakopya niyang transcription. And then, si transfer RNA, kanino dadalhin ni transfer RNA? Dadalhin ni transfer RNA yung message na yon kay ribosomal RNA or rRNA, wherein sinesynthesize ni ribosomal RNA yung amino acids into proteins. So, yun yung three types of RNA. So, messenger RNA, transfer RNA, and ribosomal RNA. Okay, so ito yung nangyayari. So, dun sa loob ng ating nucleus, doon sa loob nating cell, nandun yung nucleus. Inside the nucleus, there's the chromosome, and in the chromosome, nandun yung ating DNA. So, the DNA, nandun lang siya, doon siya nagre-replicate sa loob ng nucleus, gaya ng sinabi ko kanina. And then, yung mRNA, palabas na siya doon sa nucleus kasi dala niya yung transcription. Yung DNA na naging RNA strand. And then, ayun, si ribosomal RNA will synthesize those amino acids and then it will form into proteins. And which proteins are the building blocks of organism which carries the traits and will be physically expressed. Okay, so ito yung nangyayari. DNA, magre-replicate muna siya. After replication that happens during cell division, magte-transcribe naman siya into RNA and then it will translate into protein. So, yun yung lesson for the next reporter or presenter. Let's have Mortil, Gonzalez, and Montejo. So, Mortil, Gonzalez, and Montejo will search about the gene mutation, or defects, primarily about diabetes, Huntington's disease, Klinefelter syndrome, and sickle cell anemia. So, yung apat lang na yon. So, Mortil, Montejo, and Gonzalez will search and present about the gene mutation and defects, sickle cell anemia, diabetes, Huntington's disease, and Klinefelter syndrome. And also, ayun lang pala. And the activity will be posted. Activity number 1 will be about the DNA replication, transcription, and translation. And then there will be a quiz, a test after the presentation on Thursday. So that will be all, and thank you.